So first, thank you for inviting me. And this presentation, I named it EEG is better left alone, which is the same as a paper I published last year, already cited 97 times. And that's an argument that for standard data that's recorded in the lab, very often EEG is better left alone. And so this is our standard processing pipeline for EEG, and it's usually the same for everybody. Meaning that you have to import data. After you import data, you import the event markers and channel locations. So I'm going to go through that fairly quickly. And as I go through that, we'll see the different pre-processing stage of the data and how they can potentially affect the results. I'll skip this. So these are channel labels and locations. So usually, like 10, 20 channels in black, etc. And often you can scan locations, which we recommend. Robert Oostenveld, which was there in another symposium, his team in field trip has developed a program to scan in 3D the head. And in the EEG lab, we also have a program to import that. Hopefully, this will become standard in the next few years because it's very easy to do. You just take your smartphone, which has usually, sometimes a 3D LiDAR, the recent smartphone, and you can scan the electrode position. And that canvas paper that showed it can improve the channel location. So that's one step of pre-processing that we think is important. Then re-referencing. So very often you record data from CZ if you have EGI system or link mastoid, sometimes even nose. What's the best reference? These are different, types of reference, FZ, CZ, mastoid. Reference is the difference of potential between two sites. That's the definition of the reference. And based on the reference, you can have different ERPs. So here you see nose reference in green, link mastoid in red, average reference in blue, and the ERP looks very different. So is there a reference that's better than the others? Well, this paper on the ERN tried different types of reference here. You see average versus mastoid, and they found different effect size, but a strong overlap between the standard error here. They said, I don't think there was like a reference that was better than the other, even though the effect size was stronger here for a mastoid. So the way we try to determine what's the best pre-processing pipeline, that's our method. And that's going to be the same method for different things we'll test. First, we import the data. Then we pre-process the data using a method. So we reference, for example. Then we extract condition epochs for condition 1, extract epoch for condition 2. Sometimes we can do a different baseline, but we don't do any. And then we bootstrap 50 epochs and try to determine, and we do the difference between the two sets of epoch. And based on the amplitude of the difference and the number of significant channels in that case, we say, well, this method seems to be better than this other one because we have a higher number, for example, of significant channels using this method. So that's exactly what we did here. We tried different reference, and we saw which one has the more significant channels. And there's a lot of assumption for doing this procedure based on the volume conduction and the fact that if you have significant effect in EEG, it usually, if it's strong enough, it usually would affect most of the channels. This was, for example, all the difference reference we tested. So link mastoid, ear loads, nose, CZ reference. This is the reference in the prep pipeline. This is average reference in each lab, average reference in field trip, median reference, infinity reference, longitudinal, circumferential. And you see that surprisingly, and we still don't know why, so we're running our test, but for this specific data set, which is oddball versus standard, all the reference perform worse. So it means it's below this line. So this is the line with no reference, just the original data. And this is the performance. You see every single dot here is one data set, and the red region is the median in the center and or mean in the center and 95% confidence interval. So you see that in all cases, no reference, which means the original reference, which in this case, I think was CZ. No, it wasn't CZ. In this case, it was mastoid. They always perform worse. And we still don't know why, but just what it tells us is that it doesn't seem to be an optimal reference that's going to kill all the other ones. In that case, they all perform very well. The nose reference is worse. This is known from the literature. And the earlobes also has more noise compared to the other ones here. These are not reference which are usually very used in clinical settings for epilepsy, so it's not that relevant. So this was about the reference. This is for two other data sets. So in this one is animal versus non-animal task. It's a different original reference, and this is face versus famous scrambled face versus famous face. And again, we see that no reference really beat them all. We have less reference here because in this experiment, we really try to find the best reference. So when we recorded the data, we had all these additional electrodes on the nose, the ears, etc., to try to see what was the best reference. That wasn't the case here. These are more standard experiments, so we can't do them all. But again, we see, like the general assumption, oh, the rest reference, the infinity reference is way better than average reference. That's not the case at all in neither of this case. So this will require way more research, but it's just start to see what's the best reference according to this metric. So we can also, the next step is to downsample the data. If you have too much data, you want to downsample it. And I'm going to skip all that. Filtering the data. So filtering the data is the next step. What's the best filter? This is, so lots of times your data is offset. So, you want to recenter it because you can't see anything. So these are just low pass drift. And here we are looking again at the influence of the filter on the ERP. So this is again one of the free tasks, and we looked, it's actually the same methodology was just being used here. It's giving you a little bit more details. It says, we're going to take the ERP, and this is the ERP of the difference. And we're looking at every single subject is a gray zone. And so we're looking for a gray line. So we're looking for the amplitude in this region. And that's the same thing that was done here, by the way. We're looking at the amplitude of the difference in this region and the number of significant channel in this specific region. So that's the same thing here. The difference is that we're not testing different references, we're testing different filters. And so we see here, that's called the all-ball data set. No filter, so 0.01. That's usually what's advocated, by the way, in the ERP world. They said, don't filter your data above 0.01 because you're going to ruin the ERP. But we see here that the difference with the ERP dramatically increase if you go up to 0.5 hertz, for example. You got 60% more electrode significant when you filter high-pass filter at 0.5 hertz. And then which we also tested, so we tested on the other two data sets and find something similar. I'm not sure if we have the slide after that, but also we tested like the filter in EEG lab, the filter in Brainstorm, the filter in MNE, the filter in FieldTrip. And so you can see here, they perform very similar. And that's interesting here. You look at the filter, EEG lab filter versus the Butterworth filter of FieldTrip versus the reference here is the ERP the ERP lab Butterworth filter. And the reason this is the reference is that That's the only filter that can reliably filter at 0.01 hertz. But that's that's why we use it as the reference. But you see these ones, this one and this one and this one, they do very, very similar, even though one is a linear filter and the other ones are not. And there might be some potential numerical issue with the brainstorm and MNE. Filtering is very sensitive to the data resolution and these kind of things. There was an issue with a field trip, which we resolved with Robert. So you have to process the data in a specific way, because if you just use the FT preprocessing function in field trip, it's going to first extract epoch and then filter, which is not optimal. But we've documented all that, so now it should be fine. So filtering 0.5 hertz is kind of optimal for these datasets and for the other ones too, and it doesn't matter much which filter you use. So this was for. So this is the best filter, I think. Yeah, this is the best filter for the outboard assets. So that's the one I just showed you. This is the phase task in which there's no response, so the amplitude is much lower. And here we can see again 0.5 hertz is better. This is recorded in MEG. This is a neuromag. So even though this is EEG, this is EEG recorded in neuromag, and this amplifier actually does high pass filter at 0.1 hertz, the amplifier itself. So this might be why there's a difference between these results and these results. So the first step, so this is still, we're continuing along our processing pipeline. We've referenced, we filtered the data. Then you want to examine the data for artifacts. So these are artifacts. So you look for bad channels. And then you have automated method for finding artifacts. So here I'm going to present two, which are in EEG lab. This determined bad channels based on the pairwise correlation of channels. So it found channels which are maximally correlated with their neighbors, basically. If you have a channel that's very highly correlated with your neighbor, it usually means it doesn't contain much artifact, unless they contain the same artifact, but that's almost unheard of. So this is a good measure to determine if a channel is artifactual. So you see here we have 64 channels. Whenever you have blue lines, so this is bad data. Blue lines means low correlation with their neighbor. This is good data. And you can see the distribution is much smoother. So there's a function where you can set a threshold in the clean raw data plugin of EEG Lab to determine you want, for example, channels. I think the default is correlated 85%. And here you can see that, so this is, again, the same type of method. We take the data and we apply different thresholds for rejecting channels. You see here from 15% to 97% correlation. And this is the amount of channels rejected. And you see that even when you reject tons of channels, so here it goes up to 30%, rejecting 30% of channels. Of bad channels, you do get, a performance improvement here, about 10%, but you do lose also a lot of data. For these two, phase data set versus and the animal versus non-animal data set, even though you increase the standard deviation, but you don't increase the percentage of significant channels. Okay, so you don't increase basically the effect if you reject tons of channels. But it does actually, if you reject channels based on line noise, so which channel contain line noise instead of correlation with your neighbor, you do find that you can increase the percentage of significant channels. This was mostly visible for the outboard datasets. So the conclusion about rejecting bad channels is that at least using this metric, it doesn't help much except rejecting channels with tons of line noise. And this can improve the performance. Rejecting continuous portions of data. So this is using the ASR algorithm, artifact subspace reconstruction. And there might be other presentation about ASR and this symposium. It's widely used in the community. It was created in our lab by Christian Koch. It can use for real-time or offline data rejection. Here we're using it for offline. So we're not actually correcting the data, we're just removing the bad portions of data. And the idea is to test different thresholds. When we reject more or less data, does it increase the number of significant channels? This is what I call the gold standard. I'm going to skip over that. So the gold standard is to use humans to do rejection. And we've compared humans with machine, meaning ASR, artifact subspace reconstruction, and found that it can do as well as human. So I invite you to look at this paper. And yet, if you use it on these three data sets, you see here we reject about 80% of the data in each case, but we don't increase the number of significant channels. So we reject portions of data, but we don't increase the percentage of significant channels. And that's the main reason the paper is named EEG is better left alone, because the consensus in the community, you're going to take EEG, you're going to reject bad portions of data, and your effect will be more robust. And so here what this tells us is that even though the epochs, the noisy epochs we remove are gone, and we compute all the effect size with the remaining ones, the gain in signal noise ratio we get by removing epoch doesn't compensate the fact that we lose statistical power. So you have 200 epochs, you remove 100. Yes, the epochs will be less noisy, but no, the effect size and significance will not be stronger because now you have 100 epochs instead of 200. And so the 100 epochs, the 100 noisy one you removed was still contributing to the effect. So that's the reason what the paper is named like this. I have more slides about ICA and more slides. ICA helps a little bit, but not in a dramatic fashion. It might also depend on the metric. ICA definitely helps with the aspect of the scalp topography. So at least for display purposes in paper, it's very useful to do ICA. So you have nice scalp topography to show in the paper and not some with tons of artifact. And I know Neil will argue against ICA. We've discussed that offline. I don't agree with him, but we'll see what he has to say. This was a paper, ICA bug. There's no ICA bug. We reply to that paper. This is not controversial. ICA is still controversial after 20 years. Then there's baseline. So baseline is one of the very also interesting. Should you remove baseline of the ERP or should you not remove baseline? This is a longer discussion. I have a video on YouTube about that, but I invite you to look at. The conclusion is removing baseline, especially short baseline, is very detrimental to the effect, at least in terms of number of significant channels when you do ERP analysis. So I'm going to end here so I'm exactly 18 minutes. So I was on time, and there might not be time for much questions, but thank you for your attention.
Cool, thank you. So just before I start, thank you very much for having me here, and thank you, Arnaud, for the really interesting talk. I had a few thoughts as you were talking that I think are worth mentioning up front. One is that our results showed a similar pattern with the filtering to yours, where traditional wisdom seems to be, a high pass filter of 0.1 hertz is the best. And I think that's because previous research has shown that high pass filters can reduce the amplitude of, for example, the P3 event-related potential. But our results showed the same pattern where a 0.5 high pass filter actually performed better at detecting effect sizes of interest. And I'm wondering if that's because even though it does reduce the amplitude of the slow latency ERPs, the 0.5 high pass filter also reduces the drift and that sort of thing that can be in the signal and contribute more noise. So actually, disrupt our ability to detect the effect size. The other thing that I'd note is it's definitely not that I disagree, that I think ICA is bad. ICA is an amazing tool, very useful for de-artifacting and analyses as well. And it's incredible what EEG Lab has contributed with making that possible. My conjecture, which I'll get into in a moment, is that ICA for some electrodes, some analyses, some time windows, can imperfectly separate neural activity from the artifact. And so it does mean that you can reduce the neural signal, and that can specifically occur for one condition and not another. So I'll get into that in a moment. This is definitely a pre-processing symposium. The abstract I submitted was actually more not just pre-processing, but non-traditional EEG methods in general. So I do have some parts that are not just pre-processing, but maybe I'll just accelerate through those a bit to focus on the pre-processing aspects. So I'm just going to start by noting that the traditional EEG methods have been really informative and have really advanced our understanding of the brain over the past 100 years. However, recently developed methods have sometimes outperformed existing methods or traditional methods at detecting associations between behavior and neural activity, or have yielded important new insights. So my talk is just going to be a summary of some of the novel methods that we've used to improve our EEG analyses and reveal novel insights into brain function, just in the hope that this might inspire other researchers to explore novel methods also. So to get to the pre-processing, the most common method in the past probably 2 decades to clean the EEG data of artifacts specifically, focusing on just reducing the blink and the muscle activity. So that specific step that Arnaud was speaking about. The most common approach is to reduce artifacts by decomposing the data into statistically independent components with the independent component analysis. And then to zero out the artifact components, we identify these with IC label, for example, but a lot of people use just manual identification. And then to reconstruct the scalp space data after that artifact component or all those artifact components have been removed. So I've got an example here of how that looks, where you've got the raw or not cleaned of artifact data on the left with some blink components in there, and then the subtraction of the blink artifact component through ICA, and then on the right the data having been cleaned of blinks. One issue with ICA is that the decomposition into the source components is unlikely to achieve perfect separation of the source components. So subtracting the artifact components can remove the neural signals that are mixed into the putative artifact components, as well as the artifacts. This can alter data periods that are not affected by the artifacts. So for example, if you've got a blink artifact, using ICA can alter periods of the data where the blink artifact is not actually present. And that might be something that Arnaud or others disagree with. I'm going to present a bit of data to support, but it's still an open question, I think. So ICA's removal of neural signals can be specific to a single condition, which can lead to spurious effects when comparisons are made between conditions. That's what we've found. We have a preprint, which I guess is responding to some extent to Arnaud's publication, where we have said EEG is better when cleaning effectively targets artifacts. So what we found after a fair amount of exploration of our data and a bit of confusion as to what was happening, this explanation led us to the result where we found that if ERPs were constructed after excluding all artifacts that might be affected by blinks, ICA cleaning affected frontopolar, so the frontal electrodes that are closest to the eyes, no-go ERPs more than go trial ERPs in a go, no-go task. So this picture is just showing the epochs after excluding all blinks or anything that might be affected by a blink from the raw go trials and the raw no-go trials. And then in the middle, the topographies for the same trials after applying ICA subtract to clean the blinks. And then the difference between the raw trial topography and the ICA topography. And you can see on the bottom right there that the difference between the raw and the ICA topography is showing up in the frontal electrodes more strongly in those no-go trials than it is for the go trials. So when we look at the ERP in those electrodes, the frontopolar electrodes, FPZ, we can see in that ERP that when we reconstructed the scalp data from only the blink artifact components, there was an ERP present in the what had been defined by IC label as just a blink artifact. And that ERP differed between the go and the no-go trials. And so when we compared the difference between the go and the no-go trials in the P3, the difference was stronger after applying ICA subtract than it was in the raw data, even after we had excluded all blink epochs from the raw data ERPs. So you can see that in figure C down the bottom right there. On the left-hand side of the go mean amplitude for the P3 ERPs, and on the right-hand side of the no-go ERPs. In red is the raw data where there's a small difference, but a much larger difference for the ICA subtract analysis. And so I would note at this stage that issue, it is the strongest from what we've seen at the frontopolar electrodes. And so it might not be an issue for other electrodes or other types of analyses. But to address this point that we think is an issue, we've developed a targeted wavelet enhanced ICA method. So it still uses ICA, but it targets cleaning specifically two eye movement artifact periods within the eye movement ICA components. And so the idea here is that we're essentially trying to leave EEG even more alone by only applying the ICA cleaning to the specific artifact periods. So what this method does is it applies the wavelet transform to reduce the artifacts, but only within the artifact components, by using a progressive increase in the weighting of the wavelet transform artifact rejection strength towards the artifact periods, so that ensures the periods unaffected by eye movements are not altered by the artifact reduction method. So we found that the targeted wavelet enhanced ICA method provided artifact reduction that was equivalent to ICA subtract. So on the right there, we've got a figure that's looking at the blink amplitude ratio. So that's a measure that measures the size of the absolute blink amplitude and compares it to the non-blink periods after cleaning. And so what you want is a ratio of your blink to non-blink periods of 1, and that would indicate that the blink has been effectively cleaned. ICA subtract and a light version of ICA subtract, where only the highly confident blink components were selected for subtraction. They provide a blink amplitude ratio of around 1.1. The targeted wavelet enhanced ICA, it provided a lower blink amplitude ratio. We suspect this isn't because it is more effective at cleaning the blinks, but rather it's better at preserving the amplitudes outside of the blink periods. It also provided protection against removing the event-related potential activity when cleaning the blink components. So taking the ERPs that weren't affected from epochs that weren't affected by blinks, you can see in the middle, on the left side of that figure, the targeted wavelet enhanced ICA method within the blink artifact component contained less of an ERP than the ICA subtract method did. And so as a result, the raw data and the cleaned data were better matched after that targeted wavelet enhanced ICA method. So when we compared the cleaned data to the raw data after excluding blink affected epochs, we found a closer match between the targeted wavelet enhanced ICA method in terms of root mean squared error to the raw data compared to 6 different component subtraction methods. So that includes ICA, generalized eigenvector decomposition method, multi-channel Wiener filtering method, and a couple of others as well. So we've made that targeted wavelet enhanced ICA method available within the Relax preprocessing pipeline, which is a plugin for EEG Lab. And I've just noted the GitHub repository there. You can also find it through that EEG is better when cleaning effectively targets artifacts preprint that I've got listed up the top. Just a couple of caveats, I guess, to this result. It's dependent on our confidence in having excluded blink artifacts from the ERPs. And so this is a point that Arnaud raised actually in our discussion prior to the symposium. So we excluded blink artifacts affected epochs if they contained data within 400 milliseconds of a blink maximum, as detected by the Relax pipeline, which uses a interquartile threshold to detect blinks, which we've found to be highly effective at detecting blinks in the past. And we also excluded epochs when the reconstructed eye movement component only data contained values at frontopolar electrodes that exceeded 50 microvolts. And so blinks usually exceed 50 microvolts, and we're pretty confident that we effectively excluded the blinks. but I would very much like to see whether this result replicates independently. I'd also note that the issue with ICA and its imperfect decomposition probably doesn't affect all electrodes or analyses. So we want to be very robust in our analyses because we include all electrodes in our analyses. But if you're just analyzing a electrode, further back, PZ, for example, this might not matter. Our results were consistent across two data sets recorded with different equipment from different labs, using different tasks as well. But as I mentioned, this definitely requires replication. Looks like I don't have a lot of time left, I don't think. So I'll just skim quickly through after we pre-process. We have tried to move away a bit from traditional measures like oscillatory power and event-related potentials. We've also tried to extend beyond connectivity and complexity measures and the 1/f activity that's becoming more commonly analyzed to use an approach that's highly comparative. So there's thousands of ways to analyze time series data, and EEG is time series data. So we've used something called a highly comparative time series analysis to see out of 7,000 different ways of analyzing time series data, which types of time series analyses are best at differentiating different groups. So out of these 7,000 different analyses, we've found 405 time series features that distinguished meditators from non-meditators after applying that multiple comparison control correction across the 7,000 features. Oscillatory power and traditional measures didn't show up actually in that 405 feature list. But the stationarity of the signal did show up across a lot of different features. So it seemed like meditators showed more stable neural activity across different time windows of their data. So what I'm starting to argue for now is that there are definitely time series measures that we haven't considered yet that will likely provide a better ability to detect meaningful effects. So here's another quick example where we found that the skew of the amplitude towards more negative values was a feature that enabled prediction of who responded to transcranial magnetic stimulation treatment for depression. We've also applied something called a traveling wave analysis. So this is a bit similar to, for those who were there, Michael Breakspeare's keynote the other day. We looked, instead of just oscillations at a single point in space, like a single electrode, we're looking at how oscillations travel. And so we found in this cortical traveling wave analysis, using a 2D fast Fourier transform just across the midline electrodes, that meditators showed stronger forwards traveling waves and weaker backwards traveling waves. And this might suggest that meditators process somatosensory signals more strongly, which are reflected by the forwards waves, and generate fewer predictions, top-down predictions about what they expect will happen, reflected by their weaker backwards waves. I think I'm probably out of time here, so just to sum up, I would emphasize and encourage everybody to explore novel time series analysis methods and to try and extend beyond the traditional analyses that we apply. We can probably derive new insights by doing so. And just on a personal note, it's a lot more fun. Thank you very much for the time, and to everybody who's contributed to our research who I've listed here. Just got a short reference list there as well, and I'll end my talk there so there's time for the next one. Thank you.
Thank you very much. We have a video from our next speaker, and we have some very good comments going on in the chat. So I think what we'll do is that we will play this last video, and provided we've got time, we'll embark upon discussion at that point. Thank you very much. Up onto the our next speaker. Sam, could you please introduce it?
Hi, thank you for inviting me to speak to you today about the EEG Many Labs project and how we're using the platform to investigate emotion processing. EEG is one of the most widely used approaches to examine brain function. However, EEG studies typically have small sample sizes and consequently have low statistical power. Nevertheless, novel EEG findings continue to be generated, but replications of such studies are rare. The replication is essential for ensuring that the EEG findings are robust and credible. Therefore, EEG Many Labs aims to improve the rigour and credibility of EEG research by assessing replicability of some of the most influential EEG studies of psychological phenomena. To identify the most highly cited studies in the EEG literature, we first undertook a systematic search in the Web of Science database, where we extracted the number of citations and normalised by the age of publication. We also advertised the project on social media using the hashtag EEG Many Labs, inviting the EEG community to nominate studies they deemed worthy of replication. Through social media advertising, we also aim to identify potentially impactful recent studies that had not yet had time to accumulate a high number of citations. A more detailed description of the procedure for replication study selection is available online on the Open Science Framework. The studies were selected in a two-step procedure. Firstly, the number of citations the papers received, normalised by age of publication, and secondly, by voting by members of the EEG Many Labs community. This figure depicts the final list, which includes 27 studies, which spans topics including attention, action monitoring, consciousness, emotion, error processing, feedback and reward, learning, personality, and working memory. And the goal of EEG Many Labs is to conduct replications of at least 20 of these. EEG Many Labs has a global network, and data collection sites include individual researchers or lab groups who have volunteered to collect data for the project. The first step in the replication process is to establish a lead replicating lab for a particular study. After approval, the lead replicating lab issues a call to other labs to join the replication, listing all necessary details such as technical requirements, the expected duration of the experiment, and the planned sample size. The next stage is to make sure that the replication's methodology closely follows the original and allows the replication team to conduct a fair and high-powered test of the main findings from the original study. The replication team will prepare the materials, such as the task and analysis scripts, for the replication studies to mirror the methodology used in the original paper. Once the protocol has been internally reviewed, it is then submitted to Cortex as a Stage 1 registered report. After in-principle acceptance and prior to data collection, all methodology, materials and plans for analysis will be posted in the Open Science Framework Study Registry. Replicating labs will be expected to complete data collection within 1 year of the in-principle acceptance after obtaining ethical approval from the local ethics committee. Each lab pledges to abide by the pre-registered analysis script. However, EEG analysis scripts frequently involve manual artifact identification, correction and rejection, which introduces subjectivity to the process, and so each lab should employ the method used in the original study. This should avoid potential non-replications due to deviations in the pre-processing procedure. The lead replicating lab will conduct the meta-analysis, and replication success is defined as a statistically significant random effects meta-analytic estimate of 0.02, combining results from the different labs in the same direction as the original study. Currently, all 27 replications have a lead lab. In total, data will be collected from 168 labs from 40 countries that have committed to collecting data from almost 7,000 participants. 8 Stage 1 registered reports have been accepted, and 1 team has finished data collection, including 360 participants in 13 labs. Myself and colleagues in the School of Psychology at Manchester Metropolitan University in the UK are leading the replication of Hichak and Foti's paper, Errors Are Reversive, which explored defensive motivation and the error-related negativity. This article was published in Psychological Science in 2008 and has been cited over 450 times. In this influential study, Hichak and Foti explored the idea that error detection should prompt defensive motivation. In order to investigate this idea, they utilised the error-related negativity and the startle response. The error-related negativity, also known as the ERN, is one of the most widely studied event-related potentials. The ERN is characterised by a large deflection in the ERP waveform that peaks within 100 milliseconds of an erroneous response, and studies utilising source localisation suggest that the ERN is generated in the medial frontal cortex, and more specifically the anterior cingulate cortex, although more recent studies suggest that the ERN might also arise from alternate brain regions such as the supplementary motor area or the pre-supplementary motor area. The ERN component is largely regarded as a neural index of response monitoring processes. In order to explore defensive motivation, the authors utilised the startle potentiation, which is a robust psychophysiological measure of defensive reactivity. This refers to an increase in the intensity of the startle reflex in response to a sudden intense stimulus, such as a loud sudden noise, under aversive or threatening conditions. Hichak and Foti hypothesised that the startle responses are larger after errors than after correct responses, and that larger error-related negativity should predict greater startle potentiation following errors, suggesting that the ERN reflects a motivationally relevant correlate of error processing. So in order to test their hypothesis, the authors utilised a flanker task, which included both predictable and unpredictable acoustic startle probes delivered through headphones, while simultaneously measuring error-related brain activity through EEG and the startle response through EMG. Both of their hypotheses were confirmed. The magnitude of the startle response differed as a function of trial type, and the amplitude of the ERN predicted the degree to which errors potentiated the startle response. The authors concluded that the ERN appears to be indicative of individual variations in aversive reactions to errors. Hichak and Foti's main finding that the ERN is indicative of defensive motivation becomes our first hypothesis that we'll test in our replication. So the magnitude of the ERN will correlate with the degree of the startle potentiation following errors, whereby participants with large ERNs will demonstrate greater startle potentiation following errors. We'll also test the hypothesis that startle responses will be larger after errors than after correct responses. The statistics from the original study indicate that a total of 303 participants will be needed to achieve 90% statistical power, assuming that the effect size is half of what was observed in the original study in a one-sided test. For hypothesis 2, a power analysis indicated that a sample size of 57 participants is required. And each lab will secure approval from their local institutional review board or ethics committee to conduct the study and share de-identified data. The procedure will follow as closely as possible the process described in Hichak and Foti in 2008. As part of the broader EEG Many Labs initiative, labs will also collect resting state EEG and certain personality measures, all of which can be found on our EEG Many Labs Open Science Framework repository. The experiment was originally programmed using presentation, which has been adapted for use in PsychoPy and again is available from the project repository on the Open Science Framework. Tasks will be translated into local languages when necessary. The startle will be elicited with a 105 decibel burst of white noise, 50 milliseconds in duration and near instantaneous rise time presented 300 milliseconds after the response. The startle probe delivery will be through headphones worn by the participant during the task, and loudness will be assessed using a sound meter. The replication will be considered successful if a statistically significant meta-analytic estimate of 0.02 across replicating labs is observed, and if the effect is in the expected direction. Like all EEG Many Labs replications, we're committed to open science, and all study materials, including comprehensive details about each site, including equipment and electrode specifications, site-specific language, and participant recruitment procedures, code for stimulus presentation and data processing, will all be stored in the project repository on the Open Science Framework. To date, we have 8 labs on board across 5 countries. The anticipated data collection involves each replicating lab gathering EEG data from a minimum of 38 participants, or as many as necessary to collectively gather the data from at least 303 participants overall. Having a large international sample will provide clearer evidence into how this study design and results replicate and generalise. Our registered report is currently under review in Cortex and will hopefully be available to access soon. It's not too late to join, and we're inviting more labs to get in touch if they would like to participate in the replication of Hijack and Foti's influential study. Thank you for listening, and please do get in touch if you'd like to collaborate.
Thank you, Dr. Bland, for your talk. And we have a few minutes here to wrap up. And I noticed that the chat was quite lively. So in addition to what was presented on EEG Many Labs, perhaps we could continue some of the comments in the chat. Arnaud and Neil, if you're still here, and perhaps, I'm not sure if Robert had questions, or Bosch, and we had Rex as well. By all means.
Yeah, I think the this open source and the trend right now is very healthy for the community. We share the data, we can all process the data, people can read the papers, and they can read the response to the papers and the response to the response and make their own decision on what's right, what's not right. Because 20 years ago, somebody would say that, but data would be locked, and so they would process other data. Yeah, I think it's very healthy for the community to work in this way on common data sets. And also the fact the era of big data for EEG is here, so we have the opportunity to process more than one data set.
Something that I've been wondering about is whether we're going to end up with multiple pipelines depending on how clean the input data is, the raw data. And I'm thinking this is something that, as you say, the big data era is really going to help with. That's one potential explanation that I've been considering for the conflict between our papers, is that maybe our data, which was collected by students, is just noisier, and so the ICA separates it less perfectly. But we'll find out in the future, I guess.
Yeah, and there's also other issues of ICA in the sense if the cap moves a little bit during the experiment, ICA won't be able to extract and will distort the data, no doubt. I don't think that's necessarily the case in your experiment, but that could be the case in MOBI, for example, mobile brain imaging. So I definitely think there's some difference processing pipeline for at least mobile brain imaging and in-lab brain imaging, which is already clean data. So
I know Bosch had a comment as well. Okay, I see Robert. Great. And Rex as well.
Yeah, I think Neil makes a really good point about the quality of the data interacting with the processing that is most optimal for the data, as it's also like the feature of interest of which we try to find an effect is also interacting with the processing that we do on the data. Earlier in the chat there was a question from I'm not sure from whom it was about infant EEG data. And in my experience with infant EEG data, ICA is often problematic because it has a lot of atypical artifacts that are difficult to remove. So the data is not really a nice linear mixture. Whereas with well-consenting adult participants that sit still, ICA usually works very robust. And I think those are aspects that are important to be able to remain aware of.
Yeah, and also the fact IC label, for example, was trained, on very specific data. If you use IC label for infant, it's just wrong. It's just wrong. It just wasn't trained. It's outside. So it's going to give you some answers. Oh yeah, I'm confident. This is 99% brain. This is 99% blink. It's just totally wrong because it has never seen that data. It's not in the training set. So IC label was trained with 15,000 manually labeled data sets. So yeah, it's just outside. It's like you ask a LLM, something it wasn't trained on. It's gonna confabulate. It's gonna make up stuff.
Hi, I'm Rex. So Arnaud, thank you. I've read a lot of your papers over time. My dog is barking, sorry. Hush. But anyhow, so if you oh, okay, here's my video. My dog is barking, sorry. Anyway, so if you take this, I've been trying to get people to standardize their 19-channel EEG collection, because basically neurofeedback, that's what I do. But the point is that if you go through a standard procedure like eyes closed, eyes open, and then you have a training. So with every person that I have ever recorded, I train them in artifact. This goes from 2 years old up until 90 years old. If they watch the EEG, they can learn pretty quickly to regulate the artifacts. But then I also do the neurofeedback with these personality assessment inventories. And I've started grouping these to just do correlations for scales of anxiety, depression, borderline features, all this good stuff. But how real do you think it would be to have an algorithm that could take EEG data, the Loretta source data, and then classify it with these external measures that we have? Would
Yeah,
that
so
be,
that's the
would
holy
that be
grail.
would that
That's
be,
a holy grail for neurofeedback. I know, everybody wants to do that. So you're not the only one, or with, thousands of data sets, and it's extremely difficult. Number one, neurofeedback data acquired, a lot of it's not lab quality data. The amplifiers are usually okay, but the subjects are prepared by different people, etc. So the quality is much lower than research data. So that's number one problem. Number two problem is the noise in the metric. So you ask people if they're depressed, etc. Yeah, that's very noisy as well. So you're trying to fit two very noisy variable. And I think, there's probably some correlation, but, the main question is it usable? Because, in statistics, if you have infinite amount of data, you'll always find something. So for example, if you compare people that take 1 glass of water, that's half 1 glass of milk per day versus a glass plus 1 milliliter more of milk, and then you can find, okay, they live 5 seconds more if you take 10 million people. So this is a real statistical effect. It's real, but it just doesn't matter. So same thing for neurofeedback. For no doubt, you have 100,000 recordings of neurofeedback. Any measure you take, you're going to find some minor effect, but whether you can use these in real life is another question. So I mean, I
have a solution for you. Hold 1 second. So, see, I went to and talked with Tom Insel. We didn't even get through the first 3 minutes of it, but I trained one region, which is 3 voxels in the left precuneus in the alpha frequency, current source density wise. That is the sole target I do. I don't switch around. One target, one frequency, with the same effect in all subjects.
Yeah, and there are incredible reports in the neurofeedback community, especially for epilepsy. I mean, things where the doctor says, well, this person needs surgery, etc. And then they do neurofeedback sessions, and now they're apparently cured. And but it's very hard to control. These are not clinical trials. Very hard to control. that, you know,
Right.
it's probably real, but very hard to control. Yeah.
Well, I wrote an argument against the sham control trials because I don't really agree with how they're administered. It's really not true what they're doing. But if you were to take, let's say, because, I did some validation with MRI and fMRI using the Loretta algorithm, and it matches up pretty well. So the point is, the understanding is what is current source density and how can we translate that to EEG in real world. So what I find, because I'm also working in a jail right now in Michigan, so we reduced recidivism by 76%. They're not coming back to jail. These are substance abusers. So the point is, if you train this one area in that one particular frequency, the amplitude of the whole EEG record will drop. So let's say at baseline, there's an average of 70 microvolts on the scalp. At post-training, it will be 30 to 40. So to me,
gentlemen, sorry, we'll have to wrap up in about 90 seconds. So
Okay.
any links you can provide would, I'm sure, be appreciated. But go ahead.
Okay. So anyhow, I'm just thinking, the relations between amplitude and voltage. If you're looking at currents within the brain, it makes sense with electricity that the more efficient the current is, the less amplitude that would be required to complete a task, which is really self-monitoring, self-regulating.